Hello everyone, welcome to the next video lecture on the topic next generation sequencing analysis. So today we will be discussing how to use Bioconda Bioconda package installer to install a particular toolkit called as SRA toolkit. So in the last lecture video we have successfully installed the Bioconda and Miniconda installers in our WSL system. So today we will be learning to install how we could actually get the sequence read archive from NCBI through Bioconda. So basically this sequence read archive is actually a kind of a repository or a database that consists of all the next generation sequencing data sets that are done from different university or institutes or research centers, whatever it is, and they have actually deposited this particular read archive or particular reads or particular sequencing data they have got from their experiment into this NCBI SRA database. So basically if you are installing this particular database, not only a database but a particular package that helps, particular SRA package that helps to download this sequence read archive directly from NCBI into the toolkit, then it will be easier for us to download all this sequencing database just by knowing the annotation or the sequence ID that they are providing during during submission of their sequencing data. So basically what we need to do is that first we need to open the terminal, then we have to set up a particular configuration environment so that the Bioconda can directly install the SRA toolkit into our system. So first we have to type in a few of our codes, that is config add channels defaults. So first code is this, then you have to press enter. Okay, so it says is that warning default already in the channel list, making to a top. So basically this default is one of the channel lists that is present within Bioconda or Anaconda package. So several of the packages like SRA toolkits have to be installed after moving these particular channels to the top or prioritizing certain channels into the system first. So first we have actually prioritized this default channel at the top. Then I will press my top arrow so that the previous command that I used pops up. So this have been popped up. So I will just change this default into Bioconda. So this is the next code. Then I will again press enter. Okay, I have also moved these two channels into a top or prioritize these two channels, and then again afterwards one more channel I have to prioritize. I just press the top arrow and then replace the Bioconda by Conda Forge. Then I will again press the enter button. So in this case I have prioritized these three channels in the Bioconda package installer, that is defaults, Bioconda and Conda Forge. And afterwards I have to install the SRA toolkits, right? So this SRA toolkit can be installed either by wget or through Bioconda package itself. So when I am installing this SRA toolkit into Bioconda package, I first need to create a particular environment, that is Conda date. Sorry, there's a mistake there. You just type in this code and then press enter. So it will just take some time. Basically I already have this particular environment installed, that's why it is showing Conda environment already exists in this particular directory. So this will remove all the directory content I don't want, but if you are doing it for first time, then I prefer that you go on with the continuation of the installation. In this case I will press N in this case because I already am using this file. I don't want any changes or I don't want to reinstall in this case. So I will just show you the codes and all for you can do so that you can get it installed into your system. So I will just type it here N. So it will exit into a but I prefer that when you're typing in this particular code, right, you prefer enter and then you type in yes, so that the environment can be created there. And afterwards, once this environment has been created, so this is a particular Conda environment there, Bioconda environment. So whenever we want to use SRA tools, toolkit or SRA tool to get our sequencing analysis done, then you have to type in what a command called as conda activate SRA tools. So this is the basically the name that you are given here. So this will actually, if you have gone through the bin file that we have earlier seen in the Bioconda 3 folder, right? So there's an option called as activate. So this basically activates the particular environment that is present within Bioconda that you have created. So in this case, we are giving the name SRA tools, Conda activate SRA tools that you have just created right now. Then I will just press enter. As you can see, the base terminal has been shifted to SRA tool. So the package has been activated. So whenever this package has been activated, you can basically use it to use this particular commands within the package to get the next generation sequencing analysis done and everything. So if you want to use any command that is specific to SRA tools, then you need to activate this particular environment itself. So in order to deactivate it and move back into a the particular base terminal that you are working in, then you have to type in conda deactivate. Okay. So you are going into a base terminal then, which is probably similar to what we are using here. But I want to basically install SRA toolkit within this particular environment, that is SRA tool. So I will again activate the conda activate SRA tool. It will just activate. I will clear all the other commands that are present here. So you can see I am working within the SRA tools environment there. So within this environment, I will download the SRA tools. That is by typing conda install SRA tools. Okay. So I will be installing SRA tools in this case. So it will basically take some time. You can see the channels that you have earlier activated. It is at the top. And conda forge is a channel that we have activated earlier. So basically, it will be at the top, then followed by Bioconda and defaults. So it will take some time. So the package is now installed. You can see that downloading and extracting package, prepare transaction, verifying and executing has already been done. So as this is done, then what we need to do is that we need to configure the SRA toolkit file. So what we need to type is that we want to type in vdp config I. So basically what it does is that it opens a interactive configuration interface for SRA toolkit. So it can be used to give out all the information where you can remotely access SRA toolkit, whether it can be saved into a directly retrieved from the internet, have the internet access for the SRA toolkit, and where the data will be stored, or the repository of the data that will be stored there. And this is the AWS, and also gives access of to use a proxy and everything, and also use the user repository that you are given here as compared to the compared repository. So we will just keep this remote access on, and also we will enable the local file caching, and we will give this user repository as it is, no issue in this case. And then we will press the save here using the mouse button, and then press enter. So the save, so no changes have been saved. Okay. And then go to click on exit and then press enter. So this will actually move us into the previous terminal that you are working in. So the configuration is actually done. So and afterwards we want to see if SRA toolkit is actually installed or not. So for that I will type in fastq minus help. So basically fastq is actually a command that is present within SRA toolkit. So when we are downloading any file from this particular SRA toolkit database, it is in the form of a format of a SRA file. So what we need is a convert that SRA file into a FASTQ file. So this FASTQ dump is a command that is present within this particular SRA toolkit. As the environment is active, we can type it FASTQ dump, and in order to see what all commands that are present within this FASTQ dump command that is there, then you have to type in double hyphen then help. So if I press enter afterwards, you can see that several of the commands are there present there, right? So if you want to split the files, then you can use this particular command, that is fastq dump, then space, then type in this particular command, and then your SRA toolkit database. Then it will split up the file into paired entries and all, and also minimum read length and all. You can filter by sequence length. You can filter the database by sequence length by using this -m or double hyphen minimum read length file in this case. And so you can, there are also several functions there, so you don't need to like go into detail into that. Only you need to check whether the SRA toolkit has been downloaded. So from writing in this command, and when you are seeing this particular command list from the fastq dump file there, you get to know that it is actually perfectly installed into your operating system. So I will just clear it out. And if I want to move back into this particular like home directory or base terminal and work along, then we have to type in the name conda deactivate. This I have shown you earlier, so I will just do that again. So when you are doing that, you can see that the SRA tools Bioconda environment has been deactivated, and you have moved into a base terminal there. Okay, so I think that is for today. So in this lecture video, you have learned how to install a SRA toolkit and configure that file about using a Bioconda package installer. So see you in the next video. Thank you.